DNA barcoding. DNA barcoding is a method of species identification using a short section of DNA from a specific gene or genes. Different gene regions are used to identify the different organismal groups using barcoding. The most commonly used barcode region for animals and some protists is a portion of the cytochrome C oxidase 1 found in the mitochondrial DNA. Other genes suitable for DNA barcoding are the internal transcribing spacer rRNA, often used for fungi, and Rubisco, used for plants. The 16S rRNA gene, for example, is widely used in identification of prokaryotes, whereas the 18S rRNA gene is mostly used for detecting microbial eukaryotes. These gene regions are chosen because they have less intraspecific within species variation than interspecific between species variation. Less within species variation, and is known as barcoding gap. Some applications of DNA barcoding include identifying plant leaves even when flowers or fruits are not available, identifying pollen collected on the bodies of pollinating animals, identifying pollen collected on the bodies of pollinating animals, identifying insect larvae which may have fewer diagnostic characters than adults, or investigating the diet of an animal based on its stomach contents, saliva, or feces. When barcoding is used to identify organisms from a sample containing DNA from more than one organism, the term DNA metabarcoding is used. DNA metabarcoding of diatom communities in rivers and streams, which is used to assess water quality. DNA metabarcoding. Then Paul Herbert is considered as the father of DNA barcoding. Then methodology, sampling and preservation, DNA extraction, amplification and sequencing, and marker selection. First, sampling, data, metadata, documentation, vouchering, then DNA extraction, barcode PCR, sequencing, data validation, and databasing. Reference libraries. Reference libraries are used for the taxonomic identification, also called annotation or sequence obtained from barcoding or meta barcoding. These databases contain the DNA barcodes assigned to previously identified taxa. Several reference databases exist depending on the organism group and the genetic marker used. There are smaller national databases, FINBOL, and large consortia like the International Barcode of Life, IBOL. BOLT, launched in the 2007, the Barcode of Life data system is one of the biggest database containing more than 450,000 barcode index numbers in 2009. It is a freely accessible repository for the specimen and sequence records for barcode studies, and it is also a workbench aiding the management, quality assurance, and analysis of barcode data. The database mainly contains bin records for animals based on the COI. Then UNITE. The UNITE database was launched in 2003 and is a reference database for the molecular identification of fungal species with the internal transcribed space genetic marker region. So DIAT barcode database was published under the name RCIST diatom in 2006, starting with the data from two sources: the Thornon culture collection in the Hydrobiological Station of the French National Institute for Agricultural Research, and from the NCBI, National Center for Biotechnology Information Nucleotide Database. Then DIET barcode provides data for two genetic markers, RBCL, ribose 1,5-biphosphate carboxylase or oxygenase, and 18S ribosomal rRNA, and database also involves additional trait information of species like morphological characteristics, biovolume, size, dimensions, etc., life forms, mobility, colony type, etc., or ecological features, pollution sensitivity, etc. Then application, identification of species. Specific short DNA sequences or markers from a standardized region of the genome can provide a DNA barcode for identifying species. Molecular methods are especially useful when traditional methods are not applicable. DNA barcoding has great applicability in the identification of larvae, for which there is generally few diagnostic characters available and in association with the different life stages, larval and adult in many animals. Identification of species listed in the Convention of the International Trade of Endangered Species (CITES) appendix uses barcoding techniques is used in monitoring of illegal trade. Then detection of invasive species. Alien species can be detected via barcoding. Barcoding can be suitable for detection of species appendixes using barcode techniques is used in monitoring of illegal trade. So detection of invasive species, alien species can be detected via barcoding. Barcoding can be suitable for detection of species in a broader control where rapid and accurate morphological identification is often not possible due to similarities between different species, lack of sufficient diagnostic characteristics, and a lack of taxonomic expertise. Barcoding and meta-barcoding can also be used to screen ecosystems for invasive species and to distinguish between an invasive species and native morphologically similar species. The delimiting cryptic species. DNA barcoding enables the identification and recognition of cryptic species. The results of DNA barcoding analysis depend, however, upon the choice of analytical methods. So, the process of delimiting cryptic species using DNA barcodes can be as subjective as any other form of taxonomy. Then, diet analysis and food web application. DNA barcoding and meta-barcoding can be useful in diet analysis studies and is typically used in prey specimens cannot be identified based on morphological characters. This is a range of sampling approach in diet analysis. DNA metabolism. DNA meta-barcoding can be conducted in stomach contents, feces, saliva, and whole body. Diet analysis and food web application, delimiting cryptic species. And barcoding for food safety. DNA barcoding represents an essential tool to evaluate the quality of food products. The purpose is to guarantee food traceability, to minimize food piracy, and to evaluate local and typical agro-food production. Another purpose is to safeguard public health. For meta-barcoding offers the possibility to identify groups causing ciguatera fish poisoning from meal remnants, or to separate poisonous mushrooms from edible ones. Then biomonitoring and ecological assessment. DNA barcoding can be used to assess the presence of endangered species for conservation efforts or for the presence of indicator species reflective of specific ecological conditions, for example, excess nutrients or low oxygen levels. Then molecular systematics. Molecular systematics is the use of molecular genetics. Once again, I am going to just read the major applications. Which are the steps? Sampling and preservation, DNA extraction, amplification and sequencing, marker selection. Sampling, metadata, documentation, vouchering, DNA extraction, barcode, VCR, sequencing, data validation and databasing. Identification of species, detection of invasive species, delimiting cryptic species, diet analysis and food web application, barcoding for food safety, then biomonitoring and ecological assessment. Next is molecular systematics. Molecular systematics is use of molecular genetics to study the evolution of relationships among individuals and species. The goal of systematic studies is to provide insight into the history of groups of organisms and the evolutionary process that create diversity among species. Molecular biology has revolutionized the field of systematics. DNA evolves by mutations and is being incorporated in the DNA and fixed in populations. This will lead to divergence of DNA sequence in different species. To estimate phylogenies, first sequence divergence between the various taxa has to be estimated. DNA evolves by mutations and is being incorporated in the DNA and fixed in populations. This will lead to divergence of DNA sequence in every different species. To estimate phylogeny, first sequence divergence between various taxa has to be estimated. Then several methods are there. Then direct sequencing. Elegant molecular methods available. A lot of work. Put forward a lot of work, but provides lot of data. Direct sequencing. Each nucleotide position is a character, and the actual nucleotide that is present at that site is the character state. A character can be phylogenetically informative when nucleotide changes are shared by two or more taxa. A character can be phylogenetically uninformative when all nucleotides are the same among taxa, or when only a single taxon has a different nucleotide. A less direct method of determining sequence divergence is by restriction enzyme mapping. Restriction enzyme mapping in the sense, once again I'm going to read restriction enzyme mapping. These are the enzymes that recognize specific sequences in the DNA and cut the DNA strands. Depending on the location of restriction recognizing sites in the DNA fragments of various lengths will be generated by the restriction enzyme digest, resulting in a restriction fragment pattern. One can also determine where various restriction enzymes cut a given piece of DNA and draw up a restriction map of the stretch of DNA. The extent to which two restriction maps or restriction fragment patterns are similar serves as an estimator of sequence similarity or difference. Restriction enzymes will recognize only a fraction of the entire DNA sequence, so one will not know all the differences between two stretches of DNA. The data serve as an estimate, and because it usually involves less work, can be done on many individuals. DNA hybridization is another indirect way of obtaining estimates of DNA sequence divergence between two taxa. DNA strands are melted apart at high temperature and allowed to reanneal in the presence of DNA or another species. One species DNA has been labeled with a radioactive nucleotide. This form heteroduplex DNA. The heteroduplex DNA is gradually heated, and the amount of single-stranded DNA that has melted apart is determined by the amount of radioactive label that is counted in each fraction collected from the various temperature steps. Very similar DNA will melt at a high temperature, and heteroduplexes between diverged DNA will melt at a lower temperature. Sequence divergence is proportional to melting temperature. Phenetic approaches. DNA hybridization, sequence divergence from sequencing, restriction patterns or restriction maps. In each case, the data would be in the form of a single number indicating the similarity or difference between the DNAs of each pair of species in the study. Brown bear, polar bear, Asiatic black bear, American black bear, sun bear, sloth bear, spectacled bear, giant panda, raccoon ducks, then lesser panda. Chemotaxonomy, next one. Chemotaxonomy. Merriam-Webster defines chemotaxonomy as the method of biological classification based on similarities and dissimilarity in the structure of certain compounds among the organisms being classified. The compounds studied most are proteins, amino acids, nucleic acids, peptides, etc. Since working of the organs involve chemicals of the body, these compounds are called biochemical evidence. Then John Griffith Vaughan was one of the pioneers of chemotaxonomy. Protoplasm, end products of digestion, nucleic acid, hormones, digestive enzymes, nitrogenous excretory products, phosphagens, serological evidence, visual pigments of vertebrates, etc. The field of biochemistry has already developed since Darwin's time, and this serological study is one of the most recent pieces of evidence of evolution. A number of biochemical products like nucleic acids, enzymes, hormones, and phosphagens clearly show the relationship of all life forms. The composition of body fluid has shown that the first life originated in the oceans. The presence of nitrogenous waste products revealed the aquatic ancestry of vertebrates, and the nature of visual pigments points out the freshwater ancestry of land vertebrates. Then, serological tests indicate relationship between its animal phyla. Next, serotaxonomy, a branch of biology dealing with the nature and interaction of antigens and antibodies, or otherwise, the study of antigen and antibody reactions is called serology. The application or utility of serology in solving taxonomic problems is called serotaxonomy. The substance or protein capable of inducing the formation of antibodies is called antigens. Antigens may be proteins, nucleoprotein, lipoprotein, many saccharides, synthetic polypeptides, etc. Antigens are mostly proteins with molecular weight more than 10,000. The proteins having molecular weight less than 10,000 do not stimulate antibody production and are called haptens. Antibodies are complex proteins produced by plasma cells in the immune system. They combine chemically with specific kinds of antigens and serve as a weapon to kill or destroy the antigens. The antibodies are gamma globulins called immunoglobulins. There are 5 classes: IgS, M, IgG, IgA, IgE, IgD. Nittl was the first serotaxonomist to compare the immunochemical specificity of serum proteins and systemic purposes. An importance of serology in taxonomy: it expresses similarities and differences among different taxa, and tissue data are useful in taxonomy. It determines the degree of similarity between species, genera, families, etc. by comparing the reactions of antigens from various plant taxa with antibodies raised against the antigen of a given taxa. It helps in comparing non-morphological characteristics, and knowledge of which is used in taxonomy. Single proteins from different plant taxa are also compared by serological techniques. Then cytotaxonomy. Cytotaxonomy is the branch of taxonomy in which organisms are classified according to their chromosome number, structure, and behavior. Cytotaxonomy deals with the classification and relationship of organisms based on their detailed comparative chromosome analysis. Chromosome numbers are determined at the stage mitosis, and most cell types carry 2 sets of chromosomes. Mostly they are diploid. Then, depending on the position of centromere, chromosomes are classified into 3 categories: acrocentric, mesocentric, and telocentric chromosome. Position of the chromosome is an important telomeric character. Chromosome, cell during prophase, centromere, Q-arm, P-arm, telomere, and chromosome. Cytogenetic studies represent both structural and functional homologies among taxa based on their evolutionary conservation. It has been known that some large chromosomes have been made by the fusion of smaller chromosomes. Herbaceous plants carry large chromosomes than woodland plants. Herbaceous plants are more advanced than the woodland plant. Human beings carry 46 chromosomes, while apes carry 48 sets of chromosomes. This reduction in chromosome number is achieved through the translocation of chromosome arm between 2 acrocentric chromosomes. It has been assumed that apes were the ancestors of humans according to the cytological studies. And significance of cytotaxonomy. The role of cytotaxonomy is very important in taxonomic studies. Cytotaxonomy is more significant than physiological taxonomy because it is the comparative study of chromosomes at the molecular level. Small changes in chromosomes can be detected among the individual. Chromosomes are constituted by DNA. Variation in DNA leads to changes in chromosomes that ultimately cause variation among the individual species, genes, and everything. And numerical taxonomy. Numerical taxonomy is a classification system in biological systematics which deals with the grouping by numerical methods of taxonomic units based on their characteristic states. It aims to create a taxonomy using numerical algorithms like cluster analysis rather than using subjective evaluation of their properties. The concept was first developed by Robert R. Sokal and Peter H. A. Smith in 1963 and later elaborated by the same authors. They divided the field into phonetics, in which classification are formed based on the patterns of overall similarities, and cladistics, in which classifications are based on the branching patterns of estimated evolutionary history of the taxa. In recent years, many authors treat numerical taxonomy and phonetics as synonyms, despite the distinctions made by those authors. Although intended as objective method, in practice the choice and implicit for explicit weightage of characteristics is influenced by available data and research interest of the investigator. What was made objective was the introduction of explicit steps to be used to create dendrograms and cladograms using numerical methods rather than subjective synthesis of data. Attribute present, attribute absent may be present in some species. Next, cladistics. Cladistics is the approach of biological classification in which organisms are categorized in groups or clades based on the hypothesis of most recent common ancestor. The evidence for Hypothesized relationships is typically shared derived characteristics or synapomorphs that are not present in more distant groups and ancestors. Theoretically, a common ancestor and all the descendants are part of the clade. However, from an empirical perspective, common ancestors are inferences based on a cladistic hypothesis of relationships of taxa whose character states can be observed. Importantly, all descendants stay in their overarching ancestral clade. For example, in within a strict cladistic framework, the terms worms or fishes were used, if the terms could include humans. Many of these terms are normally used paraphyletically outside of the cladistics as a grade. There are three basic assumptions in cladistics. Change in characteristics occur in lineage over time. The assumption that characteristics of organisms change over time is the most important one in cladistics. It is only when characteristics change that we are able to recognize different lineages or groups. We call the original state of characteristic plesiomorphic and the changed state apomorphic. Wings and feathers evolve here and are an apomorphy of birds. The plesiomorphic state is for a clade is not to have feather or wings. Birds, non-Asian dinosaurs, and crocodiles. Any group of organisms is related to descent from a common ancestor. This assumption is supported by many lines of evidence and essentially means that all life on Earth today is related and shares a common ancestor. Because of this, we can take any collection of organisms and hypothesize a meaningful pattern of relationships, provided we have the right kind of information. All life on Earth is related and can be represented in a single tree: bacteria, eukarya, and archaea. There is a bifurcating or branching pattern of lineage splitting. This assumption suggests that when a lineage splits, it divides into exactly two groups. There are some situations that violate this assumption, and the other objection raised against this assumption is the possibility of interbreeding between distinct groups, which occurs at least in some groups, like plants. While such exceptions may exist, for many groups they are relatively rare, and so this assumption over holds true. Then methodology. The cladistic method interprets each shared character state transformation as a potential piece of evidence for grouping. Synapomorphs, shared derived character states, are viewed as evidence of grouping, while simpliciomorphs, shared ancestral character states, are not. The outcome of cladistic analysis is a cladogram, a tree-shaped diagram or dendrogram, and is interpreted to represent the based hypothesis of phylogenetic relationships. Every cladogram is based on a particular dataset analyzed with a particular method. Datasets are tables consisting of molecular, morphological, ethological, or other characters and a list of operational taxonomic units or OTU, which may be a genes, individuals, population, species, or larger taxa that are presumed to be monophyletic and therefore to form altogether one large clade. Phylogenetic analysis infers the branching pattern within that clade.